Okay, terrific. Well, thank you, Ben, for the invitation and the opportunity to come and talk to you all up in Michigan from balmy Florida. I just checked the thermometer and we're 35 degrees here, which I know for you guys sounds pretty tropical, but for us, that's pretty close to Siberia. So, so I'm going to talk a lot today about bacterial disease management. And really, the disease I'm really going to focus on is really bacterial spot. However, a lot of the things I'm going to talk about in regards to bacterial spot, and particularly to disease and transplant management, you really can apply to pretty much any of the diseases you pretty much deal with, bacterial speck and bacterial canker, okay? So this is part of a larger USDA funded project. We call it the Tom Spot. It's a coordinated ag project, and there's a lot of people involved. This is actually the second grant we've gotten working on bacterial spot on tomato in particular. So we've been doing this work now for going to be going on 8 years. We got one more year left in this grant. So we're really hoping the raw materials coming out will be very helpful in improving management of these bacterial diseases. For the most part, a lot of the data I'm going to speak about today actually came from our first grant. Uh-oh. Okay, here we go. Okay, a little bit of a lag there. So just so you can kind of understand where I come from, I'm actually with the University of Florida, and I work at a research center called the Gulf Coast Research and Education Center. It's located just outside of Tampa, Florida. If you're familiar with the state, it's a large research facility, and we really cater to commercial ag in our area. And again, here's kind of a map. So I always include a little Disney icon there. So if you look up in Florida, you can see UF, that's where the main campus is. You can see the little Disney ears, that's where Orlando is. And then we're here in the Tampa Bay area where the star is. So if you, whoops, sorry, hit the wrong button. So throughout the state of Florida, we have some tomato production, commercial tomato production up in the Panhandle or North Florida. But really the bulk of our tomato production and most vegetable production is really in southern Florida, kind of starting in the Tampa Bay area and moving southward. So it really is the bulk of our winter production. And right now we're pretty cold. Okay, so I people think about Florida and they think about hurricanes, tropical weather, but they don't realize that in the wintertime we're usually really, really dry, which is really its ideal time for our production. But it is a subtropical environment when we do get our fair share of moisture. And on top of that, we don't get that nice cleansing, freezing winter weather that you guys get that kind of helps you kind of cleanse that palate, if you will, of a lot of your disease issues and pest issues, right? So we just kind of go from one season and we just carry it right over to the next season. And that kind of creates a lot of unique challenges in our neck of the woods, if you will, not just the hurricanes and the tropical systems that come through. And of course, bacterial spot for us on tomato production is our major disease challenge. And with our rainy weather periods that we can get, especially in early fall and late spring, you can see really large losses and reductions in yield up to 50%. So keep in mind, I'm going to talk a lot today about bacterial spot, but a lot of the things I'm going to talk about today can really be applied to all bacterial diseases that we deal with on the foliage, including bacterial speck and bacterial canker. I get these pop-ups. In Florida, the two main diseases we really deal with is bacterial spot and speck. We really don't see canker. In my 18 years in the state of Florida, I've only seen canker twice, once in a greenhouse operation and once in a field. We've never seen persistence, and I think a lot of that really is just driven by our environment, which is a little less favorable for clavibacter than, let's say, speck and bacterial spot. So symptom-wise, of course, they get very distinct lesions on the fruit. And, for our growers, I think for most growers, this is probably the biggest challenge with a lot of these is that not only do they cause a lot of damage to the foliage, which reduces yield, but they also can get on the fruit and impact yield directly. So like I said, I'm going to focus a lot of my talk today on bacterial spot. In the Southeast United States, it's pretty much all caused by a bacterium called Xanthomonas uvesicatoria pathogens. A lot of bacterial diseases, it pretty much affects all parts of the foliage of the plant, including the fruit. The unique thing about bacterial spot that's caused by Xanthomonas perforans, it causes these very unique shot hole patterns. Let me see if I can put a pointer up here. There you go, that might be a little bit better. And of course, as those symptoms develop, you can get massive amounts of blighting and defoliation, which exposes the fruit to direct sunlight and can also cause sun scalding on the fruit and also in addition to the reduction in fruit fill. Okay. Globally, there are 4 different species of Xanthomonas that cause bacterial spot. Okay, so in Florida and pretty much most of the Southeast, we really only deal with Xanthomonas perforans or Xanthomonas uvesicatoria perforans. But I know up in Michigan and around the Great Lakes in the Midwest region, you guys really deal with perforans and Xanthomonas gardneri. Okay, so the other part is that there are 4 distinct races of this pathogen, and it kind of goes along species lines. And even though there are resistance genes available, none of them have really been deployed successfully commercially because we have these 4 different races these 4 different species that can overcome this resistance. And it just really hasn't been effective. Epidemiology, tomato bacterial spot, we start looking at sources where it can survive. Of course, you want to look at seed. It is a seed-borne pathogen. You can look at volunteer tomatoes, anything that survived from the previous season that carried over, right? Volunteer tomatoes, tomato debris can act as a major source of this disease, okay? Of course, favorable conditions are warm and humid environment. For perforans, you're talking a range from 75 to 86 degrees Fahrenheit, and anytime you get relative humidity over 65%. Gardneri, that kind of shifts down a little bit lower per se. It's probably the reason why we don't have Gardneri in Florida. It does have a little bit better success in a little bit cooler environments. Dissemination, this pathogen, like all bacterial pathogens, is predominantly moved by moisture, rain, okay? Infected seedlings, rain, heavy dew, and cultural practices, handling the plants when they're wet is really going to move this bacterium around very readily, okay? The other challenge that we've had with bacterial spot is increases, not just changes in the species over time and then the population, but also increases in resistance or tolerance to bactericides, okay? And the main one has really been resistance to copper. Okay, so what I'm showing here is a table showing 3 separate times where we've gone out and done large-scale surveys throughout the state, looking and trying to characterize the population in regards to what race was causing most of the disease, and also their tolerance to main bactericides, copper sulfate, and streptomycin. And you can see in 2006, right, we had 377 strains that we collected throughout the state from 20 different farm locations throughout the state. It was all Xanthomonas perforans, and we had kind of a 1 to 2 breakdown in regards to race 3 versus race 4. All the strains were copper tolerant, all right? And we test this by just doing little plate assays like this. This is basically a plate, and it's showing streptomycin sulfate, but we can actually take media, we actually add copper to it, and then we just spot the bacteria on there and see, do they grow, do they not grow? And that tells us if they're tolerant to copper sulfate, okay? Or we can do the same thing for streptomycin. So in 2006, we found all our bacteria were pretty much tolerant to high levels of copper sulfate, and they only around 5% were actually resistant to streptomycin. 6 years later, we repeated the same thing, smaller amount of strains, but larger number of collection sites. And again, this case, we could not find any race T3. It was all T4. But again, everything was resistant to copper, and we saw a large increase in streptomycin resistance. And in this case, it was 32% total, but 14% of that was from strains that we collected from transplant operations. That's kind of critical because transplant operations are the only places that can actually use streptomycin. We stopped using it in the field like back in the 60s when it started breaking down. And even though it's interesting that at the time when we surveyed a lot of our transplant growers, very few of them were even using streptomycin. So it was interesting that you could still find it in the transplant houses and in the field. Fast forward to 2017 and 18, and again, a much larger number of strains, larger number of collection sites. We still could find race T3, but again, it's race T4 that's driving all of it in Florida. And again, everything is pretty much resistant to copper. And again, high levels of streptomycin resistance throughout. And what we found is actually 40% of our fields that we surveyed had at least one strain that was tolerant to streptomycin. All right, so it's pretty widespread. Matter of fact, we've even changed our recommendations to transplant operations not to use streptomycin. And again, interestingly, in our surveys and talking to the transplant growers, pretty much all of them come out and said we do not use transplant or use streptomycin in transplant production. So, of course, this all trickles down into the challenges of trying to manage these diseases in field environments, right? Especially for copper, for bacterial spot. Copper tolerance is widespread, so it's pretty much ineffective in the field. And in some cases, applying copper can actually drive the disease, and you can end up with more disease than if you have done nothing. We have found that plant defense activators like Actigard are relatively effective, as well as bacteriophage and numerous other biopesticides like bacillus-based products. And I say they're relative in regards to, yes, you can reduce the amount of disease you see in the field, but pretty much all our trials are pretty consistent in that we never see a return in yield. All right? So you could slow down the disease progression. You might make the plants look a little bit better, but we have typically not seen a consistent reduction or improvement in yield to justify the use of these materials. And that's kind of a problem. So that is really what's kind of forced us to start really focusing on transplant production. And I want you to think of it this way in regards, we know, again, based on surveys and based on visiting transplant operations, that bacterial spot or bacterial diseases in these operations can be a huge challenge to manage. And think about it in regards to the tools that they're using in these operations are pretty much the same tools that we're using in the field, besides having the ability to use streptomycin, all right? The other part I like to point out to growers is keep in mind that if it's coming in on transplant and originating from seed, that is the smallest that bacterial population is ever going to be. Once it enters the field, that population only gets bigger, which is the reason why it's so much more difficult to manage it. So, again, kind of redundant. So, again, perforins, right? Xanthomonas, Pseudomonas, Clavibacter, these are all seedborne pathogens, okay? So we will talk a little bit about seed sanitation, but even with the perfect, even with good sanitation measures in regards to buying healthy, clean, certified free, disease-free seed, it's a numbers game, all right? Because to test the seed for the presence of these bacterial pathogens, it is a destructive assay. So typically what happens with these seed lots is a certain number of seed, typically a 20,000 seed or more sample is removed from that seed lot. It's crushed, ground up, and then it's tested through either PCR or direct culture plating for the presence of these pathogens, okay? So when you're talking about seed lots of up to 1 million seed or more, 20,000 is a pretty small number, all right? You probably would get better odds in Vegas than you would potentially detecting. It will, of course, detect problematic seed lots, but there's always that risk where you have a level of infestations that's just below that level of detection. And regardless of that, once you get into this type of environment, like I'm showing you here, this really is the great equalizer. When you have this many seedlings planted at these dense seed plant densities, high humidity, overhead irrigation, this is the perfect environment for bacterial spot or bacterial diseases to develop and spread, okay? On top of that, you're talking about really small plants. With this type of density, it can be very, very difficult to actually observe those symptoms until it's already flared up and it's too late, okay? So it can be really challenging to scout these and keep on top of it in a timely manner. So we'll talk a little bit more about this as well. So the other part is of course, this is kind of a nice enclosed house here, fully enclosed, but these houses can also vary quite a bit. This is kind of a typical structure that you would observe in Florida, whereas this one here is more typical of what you would see up in like South Georgia and probably further north where it's more of an enclosed structure, totally enclosed. Here, a lot of our houses are kind of, they're like a high tunnel with the sides that can be adjusted to increase or allow airflow. So again, talking about transplant and management, you know, the tools that we have available are very limited and pretty much the same tools that we have in the field. In a lot of cases, growers rely on physically roguing out diseased plants or trays of plants, right, to try to manage the disease. And there's always been a big question about, well, if we seed in transplant houses and even if we remove all the diseased seedlings or trays that we see, is it still a source of a field inoculum before we send it to the field? So this is a little bit of research that we did, and this was done back in like 2017 through 18 in my program. And what we did is we focused on 2 tomato operations. And we chose these 2 operations because they produce their own seedlings in-house, okay? And the seedling operations were not close, or at least one operation was close to a field operation, and the other one was located quite a distance away, over 50 miles away from their field operation, all right? So we went into these operations when they were starting to see bacterial disease issues and they started roguing. We would collect seedlings. Typically, we would do this like around a week prior to shipping. We'd go in and sample these seedlings, collect the strains, and then characterize them, okay? And then what we did is once these seedlings shipped out to the field sites and they were transplanted, we would come in around 8 weeks later, close to the first harvest, and do a collection again. So scout the field, collect symptoms of bacterial spot, and then we would take those isolates and actually make those we actually would characterize them and compare them. And the way we did this is through sequencing. So we'd actually take all these strains and sequence their genomes so we can make these types of comparisons. I'm not going to talk a lot in detail about this because it gets pretty involved. So really what we're looking for is we're looking for these little single nucleotide variation amongst the different strains to really show how closely related they are. All right. So this is a tree that kind of shows a genetic relatedness of these different strains. All right. And the first thing I want to point out is we got 3 different groups here: group 1, group 2, group 3. So even within perforins, we kind of talked a little bit before about the different races, but there is really different phylogenetic lineages of these bacteria. There is quite a bit of diversity within perforins. So this is really just kind of grouping them based on those 3 distinct lineages. Actually, this group 1 here is really just a reference group, if you will. Whereas these 2 here, well, actually there's the 3 groups, and really what it is they're grouped by their phylogenetic group. And then what we're using here is we're using these symbols. So if it's a purple dot, that represents a strain that came from a transplant house. If it's a green square, that represents a field. You might see a couple of green triangles, and these are kind of reference sequences for making these comparisons. And then the branch color actually represents the grower it came from, right? Grower operation A or B. So the dark blue is grower B, okay? And the light blue, which actually, for some reason, when I converted this over, actually looks more like a light green, and this one came up a little bit blue. That's actually from grower A, all right? And then we also had grower B also did some shipments from out of state locations. We actually were able to collect some strains from the fields where these transplant went out of state, and we could include these as well. So the first thing you're going to kind of notice is how things kind of group together. All right. So this would have been all strains transplanted in the field from grower A and how they all kind of group together. And you can see how grower B strains all kind of group together, transplant and field strains. Okay. It really shows, number one, we can actually differentiate the grower operations just based on the differences in the strains. And number two, you can see how the transplant strains, the strains we collect from the transplant houses in the field group together. Now, they're not going to be 100% identical because you have many generations where that bacteria is replicating from the transplant house to the field, and it's going to basically accumulate these small nucleotide polymorphisms, mutations, as it keeps reproducing, okay? So another way we can kind of present this, and this really is kind of it's kind of similar to that, but this is called a principal component analysis. So we can kind of spatially group the strains based on their relatedness. And this really goes to show where these golden circles is really the field strains, and the blue is really the transplant strains. You can really see how these cluster together, and they're actually clustered together not just based on the grower or the grower operation, but also for the distinct genetic lineages that I showed you before, okay? So really what this kind of really showed to us is that if we looked at the strains that we collected, right, in this study, 60 to 100% of the strains that we isolated from the field likely originated from seedlings growing in a transplant house. Now, I can't say they're all because they're not genetically identical because they do mutate over time, but they cluster together. The fact that the two different grower strains actually cluster closer together with those strains that came from those operations and not with each other really kind of shows that they likely originated from that transplant operation, okay? We did find a few strains were common to both field sites. They were very rare, and some field strains, and there were also some field strains that didn't correspond to any transplant strains. And really just what this really kind of shows is that you have other sources at play here. So from what we collected, it appears a transplant are really driving it, but this also shows that there's probably other sources. And the problem is that, of course, we can't collect everything, right? So it's a numbers game. But the results really stress the importance of transplants as a primary source of inoculum, okay? Seed that's coming from an organic supplier, okay? That can be a little bit trickier, okay? So typically what I would recommend, especially if it's not pelleted and you have raw seed, is to use something like a hot water seed treatment, all right? So some of the stuff I'm showing you to the right is some data, and this comes from Sally Miller's program from Ohio State University before she retired. And this really it's really what they did here is they actually had a treatment. I'm only showing you part of the data, okay? So they actually had seed lots where they split them. They did hot water treated in one half, and then they left the other half untreated. This was actually organic seed, okay? The protocol they use, and you can go online to Ohio State, and they have some great protocols there and different ways of doing this. In this case, for tomato seed, they recommended 25 minutes at 122 degrees Fahrenheit. And this is kind of what you see. So you can see the huge, so this is after they grew up the seedlings, right? And they grew them up in separate houses, the hot water treated versus the untreated. And then afterwards, they actually put them out in the field. So what I'm showing you is the end of the field season, all right? And so here you can see the dramatic reduction from untreated to hot water treated in regards to percent foliar disease. You can see that the decrease in regards to diseased fruit. You can see in regards to improvements in fruit size, healthy and mature fruit, and then also marketable yield increased as well. All right. The interesting thing about this is that when they tested this, and after they did the hot water treatment, they went in and tried to do actually testing the seed directly by taking a subsample and then testing it for the presence of bacterial spot and canker in this case, and they couldn't detect it. But yet, going into the field, you find that it's still there. So the question is, was it below their limit of detection, or were there other sources coming in? And, I would say it's probably because it was below their limit of detection per se, but there's other sources as well. But this really goes and demonstrates that cleaning the seed is a wise investment to make, all right? The other part about this is if you're growing, and another kind of a little bit of insurance for yourself, is that when you're producing your seeds for transplants, I would keep your varieties and I would keep your seed lots separated and try to put as much buffer between them as possible, all right? Because again, the closer you keep them, the higher the probability you're going to get that bacteria that starts from one problematic seed lot and gets over into your other seed lot that might be clean, all right? And I'm going to show you some data about how fast bacterial diseases spread in a transplant house, okay? Other thing that's really critical, and again, I'm not going to spend a lot of time talking about this, is sanitation and hygiene, right? Sterilizing your seedling trays, all right? Wash them, disinfect them, or steam them, all right? It's very, very critical because these bacterial pathogens can reside and survive potentially for longer periods of time than you realize, especially if you have trays that are soiled a lot, have a lot of plant debris in it. That's where they're going to survive and persist. So it's really important to make sure you clean those trays and sanitize them between production cycles, okay? This also includes your work surfaces and tools. We're doing some work right now to look on the survival of something like perforins on inanimate or just surfaces, and even on galvanized metal, we can detect bacterial spot or perforin surviving for up to 2 weeks. All right, so think about your house, and if you're doing multiple cycles, how easily that bacteria could go from one production cycle to the next production cycle if you're not sanitizing in between. Think about tools, all right, same thing. Weeds and tomato debris. There's been lots of work to show in the field how bacteria can survive on weeds, and especially for tomato volunteers during transplant production. A lot of times we can go into houses and find volunteer tomatoes where it could have been a transplant from the previous cycle that somebody pulled out and just dropped on the ground, didn't think anything about it, and there's the bacterial spot. And there's how it can bridge that going from one cycle to the next cycle. Weeds, especially any type of solanaceous type of weeds like nightshades, are really good alternative hosts for bacterial spot in particular. But there's a broad range of weeds that can act as hosts for bacterial diseases. So it's something you really need to be cognizant of. Workers, limiting worker access. Being smart with workers, and that is, if you're going to be managing your transplants, don't be going out to the field first and then coming to manage your transplants. Start with your transplants and then go out to the field, because you yourself handling plants, especially if they're wet, is a great way of mechanically transferring and moving that bacteria from disease to healthy tissue, okay? Be cognizant of your clothing that you're wearing. If you're wearing clothing that you're out in the field with, you probably don't want to be coming into the transplant house with that same clothing, because again, you run the risk of moving the bacteria from you to the plants. Minimizing any type of handling the plants when they're wet. And I'm going to go and show you some examples about why this is important. So this is some work that we did to evaluate the movement of perforins on tomato seedlings during transplant production. And again, this is a pretty typical house, commercial house, and you can see the overhead irrigation in the back. So to the right, I have some tomato seedlings to the right, and Can anybody here see the symptoms? I'll put my pointer on top of one lesion. There's another lesion. There's a lesion there, lesion there. Hard to see, isn't it? Well, the reality is early on when the symptoms develop, they are extremely difficult to see. And so we usually respond when we start seeing that foliage blight, right? And unfortunately, by the time you see it at that stage, it's typically too late, all right? And I'm gonna show you why. So we did some studies to evaluate the movement of perforants on tomato seedlings during transplant production. And a lot of this came out of conversation with growers where they're like going, well, what do we do to manage the disease? Well, when we see disease, we rogue trays, all right? And so the question was from a lot of growers is, well, if we're roguing, are we roguing enough? Some operations would just rogue the plants they saw the disease in. Some would rogue that tray. Some would rogue one tray and then another tray all the way around that symptomatic tray. And they were like, is it enough? Because even though we rogue, we'll still see disease developing. So the question was, is how fast is it moving, right? And if you see it, how much do you need to rogue potentially to get rid of the disease if you could catch it in time, all right? So what we did is we actually set up some trials, and this would be in our research greenhouse. And what we did is we set up the trays lengthwise all the way from, and I'm sorry, because this actually goes up, I forgot to break this back down into inches, but it goes all the way from where we inoculated all the way up to 210 centimeters. And what we did is we actually set zones to come in and sample over time. So we would inoculate the plants here. We would use a paintbrush just to paint on the inoculum, and we run our irrigation only in one direction, okay? So right now, the overhead irrigation is driving the bacteria, and we typically would only water once a day. In some cases, we would only water once every other day, depending on the environment, but we'd always run the boom in the same direction. And then over time, we would actually come and sample, we'd sample leaflets, non-symptomatic leaflets. We'd rate disease, collect non-symptomatic leaflets so we can actually track the disease. And the way we were able to do this is because we're actually using a strain that's tagged, it's antibiotic resistant, so we can differentiate our strain from anything else that might be in those plants, okay? So the first thing we noticed, and I think you're pretty much all aware, is that there is a big latent period or a lag time from when a plant gets infected to where you can see symptoms, okay? That's called the latent period, okay? So that period, and it comes down to where you want to cut off, can be anywhere between 5 to 10 days for bacterial spot. And it's really going to depend on the amount of disease, right? The amount of inoculum, and also the environmental conditions at that time, okay? But typically, after we inoculate, we would see within about 5 days, you would see the water-soaking lesions. And this is really critical because these can be extremely difficult to see. And typically, you're only going to see it when the humidity is high or when the plants are wet, typically early in the morning, all right? Even in a transplant facility, when that humidity is really high, you're going to be able to see these. It typically takes another 4 to 5 days or even a little bit longer, depending on the conditions, before you really start seeing these good necrotic lesions, okay? So keep that time frame in mind, 5 days to 10 days, all right? And that's after infection, before you see symptoms, okay? So this is actually showing you data that we collected. So on the left side, we're looking at disease incidence with distance, and then time is each separate line, okay? On the right-hand side, we're looking at the recovery of bacteria from asymptomatic leaflets. So we're looking, and this is basically in log numbers, right? So like a log 6, you're talking 1 million bacteria per gram of leaf tissue, all right? So log 7, that would be 10 million, right? So this is all log values, okay? And we're looking at the recovery of distance in time based on those zones we set up. And so if we go back here to the left, we're looking at disease incidence. And so it's really not until we get up to really 21 days, actually 14 days, we can just start seeing bacterial symptoms starting to show up in the zone right next to where we inoculated, okay? Reality, it's really about 2 weeks before you really can notice it and it's really taking off, okay? Let's look to the right though, where those bacterial populations are. All right, 5 days, we see nothing. That's the red line down here. 7 days, right? 1 week after we inoculated, we're already detecting bacteria almost all the way to the end of our trial, right? Well before we see any symptoms. Look at 14 days. We're just starting to see disease severity here. 14 days, those populations are exploding, okay? So these populations are what we call epiphytic populations. These are the populations that are just developing on the leaf surface, all right? They're not inside the leaf. They are just sitting on the leaf surface, and they are basically increasing as the bacteria works its way and starts causing symptoms inside the leaf, okay? So there's that big lag is really, really important. And this really also demonstrates, right, that the amount of bacteria increasing on those leaf tissues are occurring well before you see any visible symptoms, okay? And of course, this is going to have huge implications for management. So we actually did this also in a commercial seedling house. So they actually let us come in between their seasons and actually run some larger assays, okay? I know you're probably thinking brave people. So very similar, except the scale just got much bigger. So pretty much we would do groups. I think it was 1, 2, 3, 4, 5, 5, 6. So basically it was in groups of 15 trays. And we would again just inoculate the center tray, just a couple plants in the middle, and then we would just let it go over time, okay? So the first trial we ran this, and this would have been like actually early spring. It was fairly dry. Still pretty warm for Florida, but it was extremely dry. And even here, over 26 days, basically, you can see the increase in regards, and this is actually looking at recovery over time, okay? And so you can see how everything is going up over time, all right? And here we kind of looked at it. It came out, if we just did the math, it came down to about 3 inches a day we could recover. All right, that's how fast the bacteria was spreading. The second trial, things were a lot more humid, all right? So within 19 days, basically we could see the movement all the way across the trial. And so basically we're looking at on average around 11 inches a day, all right? The bacteria is moving, all right? So keep in mind that lag period we talked about, that 5 to 10 days, and you do the math, right? It's moving extremely fast with your irrigation, all right? This was another trial we did. Again, this was kind of set up in the greenhouse. So keep in mind, right, when you have all those transplants, right, it's really, if you think about it, I look at that irrigation boom, that is almost like a storm front going through your transplant house every day, whenever you water, right? And it's basically starting a tidal wave. And that tidal wave is bacteria that's being lifted from those plant surfaces and splashed to your unhealthy plant, to your healthy seedlings, right? So you're getting this surge of bacteria, right, well before you see any type of symptoms. And it's moving through the house, right? That also kind of explains why, and like in this one here, we see these little peaks, because as that bacteria gets picked up in that splash, it's going to kind of land at random places down those trays, right? So it's just that probability that we're going to detect it, okay? So we wanted to kind of look at this closer. So what we actually did is we were trying to interested is it just splash dispersal. That is just water droplets, right? Large splashing water droplets, because that's kind of what a lot of people think. So what we did is we actually kind of set up a little assay. We have tomato seedlings down here. We actually had inoculated them with the same antibiotic resistant strain. And then what we did is we just set up petri dishes with antibiotics at a specific distance away from where those seedlings were, okay? And then what we did is we didn't even move the water boom. We just turned it on for 3 seconds and then turned it off to simulate a watering event. And then because this is in our greenhouse and we know which way the air is moving because of the way the fans are oriented, these plates are actually set up downwind. So really what we're looking at is how far is the bacteria spreading, right? Just from a small little irrigation event like that. And you can see the petri dishes here to the right, and you can see all the bacteria. And that's literally just from one little event, okay? The neat part about this is that we were actually able to see how as symptoms start, again, you get the greatest amount of bacterial surge going the furthest distance. But then as symptoms matured and you start getting those really bad necrotic lesions, the bacterial populations start dropping, okay? So they're still there, but they drop quite a bit. You know, a log to 2 logs, they start dropping down, all right? So that kind of tells you that, again, perforins, again, going this distance, it really shows to us not just water droplets, it's actually because you're basically producing really small particles or aerosols that are getting picked up by the air currents and moving along. And this has been demonstrated for a number of bacterial diseases. This is probably the first real big demonstration for Xanthomonas perforans, but this has been demonstrated for Clavibacter. This has also been demonstrated for bacterial speck, okay? So think about it, aerosols in regards to, think about like when you have a cold and you sneeze, right? Those small little fine little particles that you just ejected in the air that get picked up and they just float in the air until they land. That's what we're looking at here as they land on these plates really far down here. It's not direct water droplets themselves. These are really, really micro, they're still water droplets, but they're just really, really small where they can actually get aerosolized and they stay up in the air until they deposit down, okay? And the bulk of this is actually occurring before you see those really bad necrotic symptoms, okay? So based on this, all this data, we kind of pulled together and we kind of developed the model. It's pretty rudimentary, and we're hoping to make improvements on this right now with some of the work that we're doing right now. So we kind of broke this down into low disease conditions and high disease conditions in the environment. And that's kind of what you're seeing here to the right with this quadrant, with these 4 graphs, right? So we're looking at, if you look at the columns, you're looking at cold or temperature, cold temperature. In this case, it was like temperatures below or 75 to 80 or lower, to warmer temperatures, which would be temperatures when you start getting up into the 80s, okay? There's no precise cutoff here. I would say, you know, even here I put over 80. I would say maybe even given before over 78, okay? And then we looked at conditions where you had high amount of inoculums in the rows versus low amount of inoculum here, okay? And then basically what this is showing is that as the distance increases, what is your probability of being able to find perforins, right? In regards to if you plated it out, okay? And so you can see as the temperatures increase, and as the inoculum increases, the probability of you finding it a greater distance away from the source goes up dramatically, okay? So based on this type of modeling, we came up with a kind of some recommendations for transplant operations. So we said, and part of this as well is that what we found is, you know, when you start getting cooler temperatures like below 75 degrees Fahrenheit, you still get symptom development, but the bacteria just really doesn't move that much at all. So temperature and humidity play a big role in the movement, all right? And it's some areas that we just don't really understand quite yet, okay? So what we find is, of course, anything less than 75 or 75 to 80 degrees, ZEP is really spreading about 8 inches within a week, okay? Or we found, or modeling this out, 28 inches by 21 days. So based on this, our recommendation is that you would rogue, if you were trying to rogue out the symptoms when you first see it, 1 to 2 trays in all directions from the outsource, all right, the outbreak source. So from that one tray, if you caught it early enough, you would eliminate that tray plus every tray around it, right? And this would be to have about a 95% probability of getting rid of it, okay? And again, the recommendation would be is that you do not wait more than a maximum period of 1 week after observing initial symptoms. That was based on this modeling. My recommendation would be is to get it out of there immediately. Isolate it, put it somewhere else, and until you figure out what it is, because the longer you let it sit there, every time you water, you're moving it even further. Now, high disease, and this is kind of going, 78, 80 degrees or higher. Of course, the spread is going up much quicker, right? We find spreads, perforins can spread 4 to 12 inches within 5 days, all right? So you're talking a good foot, if not more, or 1 to 6 to 5, or 1.6 to 5 feet by 12 days, okay? So think about that lag period we talked about, right? 5 to 10 days, right? So, and by 26 days, you're looking over 6.5 feet it could spread, okay? So recommendations would be once you see that those types of symptoms developing and you're in those warmer temperatures, right? You would rogue as far as 5 to 10 feet or 3 to 4 trays from the outbreak source. And I know probably a lot of you who grow transplants are going, I would lose a lot from my facility. And yeah, I realize that this becomes a really big challenge. Can you rogue ahead of it under those conditions? And I think realistically, no, you really can't. It becomes really impractical. So then we talk about using strategies like trying to change the way you irrigate, trying to increase airflow by putting additional spaces between trays. Again, you're adding buffer to try to reduce that bridge, if you will, of those other transplants to move it to the next set, okay? But again, a big part of this is early disease recognition is critical to all of this, okay? The quicker and earlier you see it, and not before you start seeing the blighting leaflets. If you can catch it, the earlier you can catch it, the higher probability you would be successful in being able to rogue it out, okay? But just keep in mind that it really is your irrigation environment that are driving this, okay? And again, the epiphytic populations and that 5 to 10 day latent period, right? So before you see symptoms, right, you got to realize that the bacteria is already there. So wherever you see symptoms, you need to realize that it's already moving, right? So that's the reason why you need to be aggressive when you rogue it out. Try to rogue it out. Okay, so we've done some more research to try to look at this a little bit more. A lot of what I showed you, previously with this work, putting out the Petri dishes is kind of nice because it kind of gives you just kind of a quick visual, but this is kind of a passive sampling strategy. You're kind of you're basically waiting for gravity to pull that particle down and deposit it on the plate, all right? So we can kind of qualitatively say, hey, it's getting this far, but we can't tell you really how much is up in the air that we're not really collecting because it's just not settling out or it's going much further, right? So we're working with a colleague over at the USDA, Walt Mahaffey, and he's kind of developed these active samplers, and they're called cyclonic air samplers. And these are samplers that have been around. This technology has been around for a long, long time. This is nothing new. It's just because in the past, to try to buy these types of instruments, you were talking tens of thousands of dollars to buy one of these to do this type of sampling, active sampling. Whereas he's using a 3D printer to print these things out for, $5. Okay. So it allows us to do a lot more work to really try to look at this. And the neat part about this as well is that as we're sampling here, we're actually able to keep the bacterium alive to say, okay, is it viable? So it's not just doing a collection, doing a PCR and saying, can we detect it? And not knowing how much of it was really viable. This is actually telling us we're able to collect viable bacteria so then we can kind of follow our strain and we know it's really alive. So this is some work a couple students are working on right now, Renzo Ramirez and Garrett Giles. So what you're seeing here is just disease, some data that we've only have a couple of years, we've been working on for the last 2 years, and we're hoping a lot more will come out of this. So they kind of set up the trials pretty similar to what I showed you before. These groups of trays, you got your seedlings, and typically we would go in and just inoculate a couple seedlings within that. And we would inoculate the seedlings at 2 different levels, kind of a log 4. So in this case, you'd be talking about around 10,000 colony formees or bacterial cells per mil versus 1,000,000 bacterial cells per mil when we applied it to the seedlings. And then we use overhead irrigation. Again, try to just go in one direction, but we're not really looking at movement here. We're just trying to look at the bacterial populations over time. And then what we did is we came in and we measured disease incidence, epiphytic populations from asymptomatic leaflets like we did before. And then we also looked at the recovery of those aerosols using this active sampling. And we did this before and during irrigation. And then we actually included an additional control for the cyclonic air sampler because we're kind of concerned. So basically what we had is we had a tray that we inoculated, but we didn't use a cyclonic air sampler because we're kind of worried that if we're using that vacuum to vacuum up the bacteria, are we changing the disease progression, right? Are we sucking up so much bacteria, which would be kind of cool if it worked, that you're preventing disease from developing or changing the way it develops. So we included kind of a control for that. So this is looking at number of disease plants. So these were 200 cell trays. And so it's based on the number of plants per 200 cells. And this is kind of looking at disease development over time. And so we're looking at here log 6. So this was the treatment where we just did log 6. This would be in the treatment with log 6 with no cyclonic sampler. So really what we saw is the disease progressed exactly the same, whether we use the cyclonic air sampler or not. So that wasn't changing the way the disease progressed. And then you can see here the log 4. And you can see it's much less disease. So the disease progression is much slower because you have less inoculum to begin with. And these were actually non-inoculated trays. And even though we had them spread out quite a bit in the greenhouse, we still saw disease starting to develop in these. And again, it's probably coming from this log 6 because it's moving around. And I'll show you why that probably is. So this is actually looking at the epiphytic populations from asymptomatic leaves, okay? So again, if we look at our treatments with the log 6, you can see those populations get up much higher and much quicker than where we had lower populations to start with, okay? Again, this kind of re-emphasizes that model I showed you before of having high amounts of disease versus low amounts, or high amounts of inoculum versus low, how that drives that disease development. So these are looking for asymptomatic seedlings, okay? So those epiphytic populations are really, really high. And here you can see on our non-inoculated, you can see they're really low, so we didn't detect anything to begin with. But around week 3, it really jumps, okay? So this is actually coming from the cyclonic air samplers, okay, themselves. So this is active sampling. We're vacuuming the air to capture the bacteria and to quantify it. So this is looking at our non-inoculated control. And what this is several samplings over time, and that data kind of merged together to make it a little bit easier to see. So we're looking here at our non-inoculated control. The pink on the left would be before irrigation, and then the blue is actually during irrigation. Okay, so as that boom is going across the tray, we're coming in and actually sucking up, actively sampling the air right behind it. Okay, so here's our non-inoculated control. Here's our log 4 population that we started with, and here's log 6. The interesting part, yes, when we irrigate, we do see higher levels. But even before we even turn on the overhead irrigation, right, we're able to get aerosols of the bacteria in the air. So it is already moving, and it probably explains why, even though we have a non-inoculated control, we're still getting the bacteria moving around and landing on those plates, on those trays, and establishing in this environment, okay? So any questions so far? So hopefully this really helps you understand why bacterial diseases are so difficult to manage in a transplant environment and even in the field. Because if you think this, right, in a transplant house where you're doing overhead irrigation, the data I'm not showing you is we've done the same thing in the field. And when you start looking at a high volume tractor sprayer, yeah, you get a giant cloud of bacteria that's being blown around with the sprayer. So, any questions so far before I move on?
Gary, I just mentioned real quick, I have a lot of questions, but I know you're about to get to some really good stuff. Um, Folks, if you do need to leave, I just in the chat put the survey link if you need those pesticide credits. This is this is simultaneously really interesting and really, I don't know if scary is the right word, but I'm curious to see what you share next, Gary.
Okay. Yeah, I'm sorry, I lost track of my time with answering questions here. So, okay, so what we've done so far is what I want to try to go in is like, okay, the bacteria is there, it's moving, you're roguing. What can you do to try to manage it in regards to bactericidal materials to apply? So we've done some work to evaluate the integrated use of bactericides, ActGuard, and other non-copper alternatives. This is a huge list of materials that we've looked at. We actually evaluated 13 different products across 8 trials, transplant, seedling trials, either solo or kind of integrated into different types of bacterial spot management programs. And sorry, this is advancing on its own. So really, the 3 I really want to point out are really the 2 is really ActiGuard and Quell. So this is really another slide showing of all the products that we trialed, at least against bacterial spot, ActiGuard and Quell, which is it's a copper octanoate. It's really a copper soap. So it's really a low level of copper compared to Coside. For whatever reason, worked the best. And I'm sorry, my slide keeps wanting to advance. So here's our untreated control. This is really looking at disease severity. This is just one of 8 of the trials we did. This is phosphoric acid. This is phosphoric acid with bacteriophage. This is copper hydroxide. So this would be our Coside 3000. This would be the phage on its own. So this would be tannose plus phosphoric acid plus phage. Quinoxyphen, which is Quintec, which I loved here. It's not labeled for tomatoes, but it is labeled for peppers. So if you're looking for an additional management tool for pepper bacterial spot, you should take a look at quinoxyphen. And then we start getting into combinations where we got copper octanoate or ActiGuard kind of mixed in with these other ones. And really the take-home message came when it came on products on their own. ActiGuard, one application of ActiGuard about a week after seed germination, kind of carried it throughout the entire season. We have a number of operations that have kind of adopted this. And then copper octanoate, which is Quell. I apologize. I think there's actually another commercial product that's labeled, and it's also copper octanoate. I can't remember the name of it right now, but there's another product available other than Quell for use in greenhouses for bacterial spot management. And these products worked extremely well. The interesting part is there's also a product called Agris with silver nitrate. It's actually a product they use for coatings. Okay, and it actually has silver in it, and it's very bactericidal and worked very well in our assays. I didn't include it here because it's not registered and it's extremely expensive. So there's no need to really talk about it any more than that. The other part is the chemical performance of these same products we actually tested in the field. And like I told you before, yes, they work to different degrees, but you just never see an improvement in yield. And the reality is that you might see maybe a 5 to 10% reduction in disease severity, but you just don't see the recovery in regards to yield. So it kind of comes down to, does it really make economic sense in some cases? But in the transplant house, at least these 2 made a profound difference. So just kind of wrapping up with just a summary. And, I know you guys probably have a lot of questions, and I'll try my best to answer them. Again, starting with healthy, clean seed, I think that's pretty obvious. However, keep in mind that regardless of what you do with the seed, it's a numbers game, all right? And unfortunately, there's a lot of seed, and you can't test every one of them. So the probability of that disease being there below your limit of detection, or the limit of detection being used to test the seed, is pretty high, okay? But I think overall, I think seed companies do a really good job. And I've been to a number of places where seed production is occurring. They really go out of their way to produce the seed and keep it clean and test it and produce it in areas where tomato is not being produced, all right? To try to minimize that risk, but it's always going to be there, unless maybe if we produce seed in outer space, I don't know. Keep those separate, those different varieties and seed lots separated. And again, try to create as much buffer between them as possible. Like you saw, the aerosols are gonna move, all right? But distance, the greater the distance, you're going to lower the probability of that bacterial aerosol finding a new home, okay? Sanitation and hygiene, we talked a little bit about that, the importance of it, making sure everything is cleaned before you start, but even between production cycles, okay? So you don't just carry one disease problem from one transplant production cycle right into the next production cycle, okay? Scout and rogue early, all right? Like I showed you, right? The environment and your overhead irrigation is really driving everything. The sooner you capture the disease, the faster you can get it out. I see it a lot of growers who have actually changed the way they put their seedlings out. So here's showing kind of blocks with these rows in between. A lot of growers have actually gone up and tried to maybe limit themselves to like 3 rows on like speeding or like rail systems, and in putting like a 2-row break. Again, improves the airflow. Yes, you have to be careful because your edges are going to dry a little bit quicker, right? But it also allows you access to get in there to scout closer as well, okay? Also minimizes maybe how much you have to handle those plants if they potentially are wet when you're trying to move them to get inside to try to look at them as well. So if you have the space, use it to your advantage. Okay, overhead irrigation. It would be great if everybody can move away from overhead irrigation. I know that's not possible for everybody, but if you could go to an ebb and flow or a capillary mat type of system, they do work. There's been some great studies that have shown they can really reduce the spread of bacterial diseases, especially on foliar like bacterial spot, speck, and canker. But it is a huge change into the production, and I know a lot of growers don't like it because you don't get the same type of air pruning of those roots, and that kind of leads to other problems in regards to damaged roots, and so you can get more more transplant or seedling losses in the field during transplanting. So a lot of growers just don't like that system, so they still stick with overhead irrigation. If you are irrigating, do it sparingly, only as needed. Try to make your irrigation in the morning where those plants can dry out quickly. Okay, I'm not saying do it at 6 o'clock in the morning when humidity is already high, but doing it at like 9 or 10 o'clock in the morning where everything's kind of warming up, your air is dry, it allows plenty of time for those seedlings to dry out during the day. You don't want to go into wet plants in the night because that would be even more ideal for bacterial spot. Low water pressure. How low? We're working on that right now. Operations can differ dramatically in their water pressure. I've seen some operations that are watering at 40 psi all the way down to 10 psi. And I get asked this question about that and nozzles. Those are the things we're trying to look at right now. My best advice is it's kind of common sense. The higher the pressure, right, the more spray and the more aerosols you're going to generate. All right, so the lower you can go with the least amount of splashing is always going to be better. So we're trying to do different pressures, and then trying to look at different nozzle types, whether you use a fan versus kind of a soaker type of nozzle. And again, some of this might be even looking at what you can do to bring those nozzles down closer to the plants so you can run lower pressure and still get better coverage. All right, so those are things we're trying to look at right now. All right. Apply effective bactericides. If you're going to use them early. And I think sometimes that's where it can be problematic because some growers will use higher pressures for when they're making their bactericide applications. And I think that's something we just have to be aware of is there's going to be a trade-off there. Yes, you might be improving coverage and making it easier to apply it, but if it's not very effective, you could also be moving the bacteria around more than what you realize, okay? And then between production cycles, that sanitation, hygiene, we talked a little bit about that. Volunteers and weeds, I think that's again another kind of common sense. I didn't include that in here because we're actually doing a couple of studies at a couple different transplant operations where we're going out there and sampling weeds. And of course, we're doing this in the field. It's amazing how many weeds, common weeds, where we can pull bacterial spot in a transplant operation on the outside. Now, whether it's getting back to the inside of the transplants is the big question. But just thinking about rain events on the outside that potentially could aerosolize that bacteria, allow it to get inside, especially in facilities that have sides that can be raised and lowered to improve air exchange, you can't rule it out again. All right. There's a lot of people who are involved in a lot of these projects. So I'm really kind of capturing a lot of research that's been done over the years. So with that, I will open this up to any questions you might have.